To start off this lecture, we're going to talk about bacteria cells and how they divide through binary fission, and then how their DNA is copied. They have DNA replication taking place in their cells as well. And I just wanted to show you this table, and you should recognize pretty much all these molecules because these are the molecules, these are the enzymes that assisted in DNA replication in eukaryotic cells. So again, we're bringing this up for like the 20th time this school year, is that when you see common molecules in different species, it indicates a common ancestor. So the genes that code for these enzymes were selected for over time. They are passed down from generation to generation, and that's why we have generations today that have these genes coding for these different enzymes. So here's a good time to talk about how they can be tricky on essay questions. And they will give you a specific organism. So they'll say, talk about a protein that assists in DNA replication in bacteria. But I could pretty much say, talk about an enzyme that assists in replication found in the cells of an octopus. It doesn't matter which organism I say, the enzymes are pretty much the exact same enzymes in all organisms. So if it says discuss some enzymes that are involved in bacteria cell DNA replication, pick the big major players. What does helicase do? Whatever it did in your cells, it's going to do the exact same thing in a bacteria cell. Pick DNA polymerase 3. It's the one that matched up bases and elongated the strands of DNA whenever DNA is being replicated. Topoisomerase, you can see it in there. We've talked about it in our replication. Okay, so it doesn't matter the organism. If it's asking you to discuss enzymes that are assisting in DNA replication, just pick some of the ones that you remember best and talk about those. Also, this is a good time to indicate if they tell you to talk about 3 enzymes, and let's say you think, oh, I'm going to talk about 4, maybe that'll get me some bonus points. They will not grade the 4th one. If it says talk about 3 enzymes, they're only going to grade the first 3 that you mention. So always make sure that you pick 3, and those are your best 3 that you know the most about, and talk about those. Now, let's look a little bit more into DNA replication of prokaryotic cells. And you can see one big difference is that they have circular DNA, not linear DNA. We see linear DNA in all eukaryotes. Again, that's an indication of a common ancestor to all eukaryotes. It's because they all contain linear DNA. And you can see some of the major enzyme players that we talked about here. So we have single-stranded binding proteins keeping the strands apart. We have helicase. We see there's an RNA primer put on. So it's the exact same equipment, pretty much replicated the exact same way. It's just that the DNA is circular instead of linear. Now, before a bacteria cell divides, it's copying its DNA. Bacteria cell division is called binary fission. So DNA replication in bacteria cells happens prior to binary fission. And this type of cell division is not for healing and growing, like we've been talking about, because bacteria are single cell organisms. So this is a mean to reproduce. The reason why they split in 2 is not to heal and grow. They're trying to make more individuals. They're just trying to reproduce. Now, the daughter cells are going to look identical. Here's our daughter cells. They're going to look identical to the parent cell. So we say that this type of reproduction leads to no genetic variation because the parent cell is going to look or have the same genetic information as the daughter cells. And then I liked this diagram because I wanted to show you 2 things. First of all, bacteria cells only have 1 chromosome. So let's indicate that. They have 1 chromosome, and it's circular. But I want to point out that they have extra pieces of DNA, and we're going to talk a lot about them in January when we're in the molecular genetic unit. So they have extra pieces of DNA that are also circular, and these extra pieces of DNA are called plasmids. Now, when a bacteria cell divides, it'll copy its regular 1 chromosome that it possesses, and it'll also make copies of the plasmids. So each of those daughter cells are also getting a copy of a plasmid. So I just wanted to point this out because you see basically kind of 2 pieces of DNA in here. You have the original chromosome, and then you have some extra DNA, which is called a plasmid. You might recall me mentioning that I was putting DNA replication into this unit about cell division, since The cell goes through the cell cycle, and in the S phase, the DNA gets replicated. I decided to show you what DNA replication looks like. And again, one reason why I did that is because it was normally in a molecular genetics unit, just basically a unit about DNA. And that's a really big unit. It was a really hard unit, and students were getting replication and transcription all mixed up. So I moved replication to this unit. Now, I'm also moving something from that unit. I'm going to teach you how we can make copies of DNA. Just like we need to make copies of papers, we need machinery so that we can make a large amount of DNA, that we can copy the DNA. Instead of saying copying DNA, I can also say that we're cloning DNA. And there's two basic ways that we can clone DNA. But first, let's talk about why on earth would we need large samples of DNA. Well, if you remember, I indicated that I had my DNA sequence. So I sent them some samples of my cheek cells. Basically, I spit in a cup and I sent it in the mail to them. And what they'll do is they'll take the DNA out of my cells and they will copy it. They'll make many copies of my DNA. And they do that so that there's tons of DNA from me to analyze. So we will want to make large copies of DNA whenever we're trying to do research on specific genes. If we want to sequence someone's genome, we have to make multiple copies of it. If you send in a sample of your cells of your DNA for genetic testing for disease, they'll make multiple copies of it. And if we can get some DNA from a crime scene, then what we want to do is make multiple copies of it so we can perform a lot of tests on it to see if we can figure out a person who was at the crime scene. So scientists had to develop ways to clone DNA or replicate DNA, or I can also say copy DNA, and we also can say that we're amplifying DNA. Amplify means to make bigger, so we're trying to make our sample of DNA bigger. So amplifying DNA is just another term that we can use. So here's the two ways that we can possibly copy DNA. The first way is using bacteria, and this was basically the old way of trying to make multiple copies of DNA. So bacteria divide by binary fission, and they divide very quickly. So if we could take some DNA, so let's say there's a gene that you wanted to make multiple copies of, and basically here's that gene right here. Remember, circular pieces of DNA that are extra pieces of DNA in a bacteria cell are called plasmids. So this is why it's called a plasmid. It's an extra piece of DNA that we can put in bacteria cells. So if we can insert a gene into a plasmid, and we can take this plasmid and we can put it into a bacteria cell. And then we take that bacteria cell and we put it on a growth medium, an agar plate. Then that bacteria cell is going to divide into 2. And then it's going to divide into 4. And it's going to divide into 8 and so on. And every time it divides, it copies its DNA. So we're going to get a ton of copies of this DNA. We can then pull the DNA out of the bacteria cell and we can analyze it. But again, this is kind of the old school method of copying DNA or cloning DNA, is using bacteria. And now we just simply built a machine that's going to help us copy the DNA. So this is called a PCR machine. Now what we're going to do to copy DNA is that we're going to basically put materials into this little tube. So in goes the materials, and I'm going to go through those materials in a second. And then we're going to take the tube and we're going to put it in the PCR machine. So in goes the tube. And you can see we can load a lot of tubes into the PCR machine. And basically what the PCR machine does is it just heats up, it cools off, and then it heats up a little bit, and that's one cycle. And the heating and cooling is what is going to trigger this DNA to copy itself. And then we're going to repeat the cycle. So we copy more and more and more. So eventually, whenever we're done, we're going to have billions of copies of the DNA just in the test tube that we take out of the PCR machine. So let's take a closer look at this technique, the PCR technique. Let's first talk about the materials that we're going to put into that little tube that I showed you up there. That's going to eventually go into that PCR machine. Well, if we're going to make some DNA, obviously we need a large supply of the nucleotides that make up DNA. So we need nucleotides that contain adenine, guanine, cytosine, and thymine. So we're going to put a really large supply of nucleotides into the test tube. Now remember, the reason why we're doing this is we're trying to copy a piece of DNA. So maybe this was DNA from a crime scene and we want to make multiple copies of it. So we have to make sure that we put in this segment of DNA that we want to copy. Now, just like when we replicated DNA, we first had to build some primers so that DNA polymerase hooked on. So we can actually engineer primers. So we're going to build primers that will actually kind of section off the parts of the DNA that we want to copy. So if we have a long piece of DNA, so let's say this whole thing is DNA, and we just want to copy specific parts of it, if we just want to copy this part, then we have to put a primer in there that is complementary to these nucleotides here and these nucleotides here. So we're going to have a primer that will bind right here to those DNA nucleotides. So we have to make primers, engineer primers. They have to be complementary to the ends of the DNA that we want to have copied. So that's why these primers are going in and showing you the primers. And the primers are made of RNA nucleotides like A's and U's and C's and G's and so on. So we have to put in a large supply of primers that are complementary to the ends of the segment that we want copied. So basically, here's kind of, we want to copy from here to here. So those primers are going to anneal or attach to the ends. Now finally, we're going to use a specific enzyme. We're going to use DNA polymerase, but we have isolated this DNA polymerase from a bacteria cell that lives in environments that are super hot, like almost boiling. They're hot springs bacteria. And so this DNA that we pulled out of the specific type of bacteria cell that lives in hot springs, it's called Taq polymerase. So we're going to use Taq polymerase. Taq polymerase is really just DNA polymerase that's removed from that cell. And the reason why we must use this Taq polymerase, a special DNA polymerase, and not our own DNA polymerase, because remember, the PCR machine is going to heat up to really warm temperatures, and proteins can denature. We need a DNA polymerase molecule that can function in really hot temperatures, and it does in those bacteria cells that live in those hot springs. So let's show you exactly what's going to happen to the DNA molecule that we need copied, that we placed into that test tube. So what we're going to do is we're going to take the test tube, we're going to put it in the PCR machine. And so this is basically what's showing you here is this is what's happening in the test tube. This is the DNA to copy. So let's put, here's the DNA to copy in the test tube with those other materials. And this is what our PCR machine is going to do. The first thing, and you see it here with the number 1, that PCR machine is going to do is it's going to heat up the contents of that test tube to 95 degrees Celsius. This is almost boiling. Now, remember that high temperatures breaks the bonds in proteins. It also breaks the bonds in DNA, and specifically, it breaks these H bonds. Remember, there's H bonds. Let's put an H bond in there, holding the 2 strands of nucleotides together. So when you heat up the DNA to 95 degrees Celsius, it breaks the hydrogen bonds. It breaks the H bonds, or we can say that it separates the strands. So we can see the 2 strands are now separated, and they have them color-coded for you. So here was a black one, and here was a blue one. So here's the black one, and here's the blue one. They have been separated. So we say that we're heating it up. We are actually denaturing. So we heat up to denature the DNA. Or we can say that we heat up to separate the strands. The next thing the machine is going to do is it's going to cool off the test tube to about 55 to 60 degrees Celsius. And what that does is it allows these primers. Remember we said we built primers that would be complementary to the bases here on the ends of the DNA segment that we want to copy. So here's a primer, and those RNA bases are complementary to the DNA bases here. That's at the end of this DNA segment that we want to copy. So when we cool off the test tube, the hydrogen bonds can now form between the primers and the DNA molecule. So we're getting some H bonds forming as the primers bind to the complementary DNA strand. Now, we have a name for this. We say that we cool to allow the primers to anneal. So it's cooling, or it cools to anneal the primers. That means allows the primers to bind to the DNA. Now remember, DNA polymerase operates at high temperatures. It's not going to operate at 55 or 60 degrees Celsius. So that's why the machine now has to heat up to a warmer temperature, 72 degrees. And here it is. This is our Taq polymerase. This is DNA polymerase. And remember, it can only bind to a 3' end of a nucleotide of a primer. And we have that with the primers attached to the DNA. So a Taq polymerase bound to the 3' end of the primer, and it is elongating that strand. It's copying the DNA. And this one bound to the end of this primer here, and it's copying the DNA. So you can see that I now have double-stranded DNA. We just basically made a photocopy of that original segment. So that third step, we heat back up the DNA so that the DNA polymerase will elongate, or sometimes we say extend. And that simply means adds these DNA nucleotides to extend or copy the DNA. So basically, by the time we get here, one cycle, so let's put one cycle, is done. So we had a heat, cool off, heat, and we made 2 molecules of DNA from one. So that first cycle is done. The machine is not done. It's going to repeat cycling, hot, cold, hot, 30 to 40 times. It depends on how much DNA you want. So I don't like the fact that it says number 4 here, heat back up to 95. We're actually back to step 1. Heat it back to 95. It's going to separate this one. So this one is separated. This one is separated. You can see it separated down here. So then we cool it off. The primers are going to anneal. We heat it up a little bit more so DNA polymerase can extend the strands. So then we're going to end up with 4 molecules of DNA, because this one will end up being a double helix. This one will end up being a double helix. This one will be a double helix, and this one will be a double helix. Now think of the numbers that we can get. If every time we're doubling the number of DNA molecules, by the time this machine cycles 35 times, from that 1 molecule of DNA, you end up with 68 billion copies of that DNA segment. So we are copying DNA. We're replicating it through a process that's called PCR. Now I just want to show you a couple more diagrams related to PCR, and hopefully you kind of understand what's going on. It's just a different way of showing you what's happening here. So again, we have the 3 steps. We have the heating, we're going to have the cooling, and then we're going to have heating up again so we can extend this strand of DNA nucleotides like you see right there. So again, the heating up denatures the DNA. It separates the DNA strands. When we cool it off, hopefully you can recognize what these would be. These would be the primers. And we can see that primers are binding here. So here's a primer. And we can see that the primer is binding here as well. Whenever we heat this back up to about 72 degrees Celsius, then let's attach our DNA polymerase, and it's going to extend, or it's going to elongate this strand of nucleotides, copying the DNA. So we have double-stranded DNA here. So here's a DNA molecule, and we're going to end up with double-stranded DNA here, because DNA polymerase is going to extend this strand of nucleotides. So we went from 1 molecule of DNA to 2 molecules of DNA. This is just a different way to show you what's happening in that tube that it's been added to that PCR machine. But again, think about what we had to add to that PCR tube. We had to add the DNA to copy. So here's the DNA that we wanted copied. We had to add primers that match the end of the segment of DNA that we want to copy. So we really only wanted to copy from here to here. So we made primers that were complementary to basically the ends of these DNA molecules. And then we have to add a bunch of nucleotides, and you can see them in the diagram. And then you have to add that Taq polymerase. And you need to be familiar with all the materials that we need to add to a PCR tube so that we can copy DNA or clone DNA or amplify DNA. There's different ways to say it. The reason why I wanted to include this diagram of a PCR is to highlight this idea that we don't get a copy of this segment of DNA that we're trying to copy until we get to cycle 3 of the PCR. Now remember, whenever researchers are using PCR, they usually want to amplify just a segment of the DNA. So here is the DNA molecule here. And they want to amplify or copy this region right here. And when we look at the diagram, we can see that we do not get just this area right here until we get to cycle 3. And then we finally have 2 copies of that segment. There's a copy and there's a copy. So in cycle number 3, we do finally have 2 copies of the DNA segment of interest. When we get to cycle 4, you can count and you can see that we have 1, 2, 3, 4, 5, 6, 7, 8 copies of that segment of interest. Now the reason why we do not get that segment of interest until we get to cycle 3 is because you have to remember that DNA is a really long molecule. So here we have denatured or split open a DNA molecule, and it's just showing you part of it, but the DNA is a really long molecule. So let's go ahead and show that. Now remember that we have engineered primers to bind to the ends of the segment that we want to copy. So the primers are bound, and you can see DNA polymerase. It binds to the primers, or that 3' end of that last nucleotide of the primer, and it's going to build a new sequence of nucleotides. And it's going to keep going until it goes to the very, very end of the DNA molecule. Same thing is going on this bottom strand, is that Taq polymerase is binding to the 3' end of a primer, and it's extending a new strand of DNA nucleotides, and it's going to go to the very end as well. So we can see that if we analyze these 2 molecules of DNA, because we have double-stranded, this is double-stranded, I'm going to put DS, that's double-stranded DNA. Here we have double-stranded DNA. It's not exactly what we want. And again, we will not get copies of just the segment that we're trying to copy until we get here to cycle 3. Another reason why I wanted to include this diagram is because I like how it shows the 3 different temperatures that are utilized in this PCR technique. So again, we have a really high temperature, and that is denaturing. So let's write denaturing that DNA molecule, breaking apart those hydrogen bonds. Whenever we drop that temperature, we're going to allow those primers to anneal. So let's indicate during this time the primers are annealing. And hopefully you can tell me what would be going on this 3rd temperature. That's whenever we raise it up a little bit to a temperature where Taq polymerase is functioning, and it is extending this strand of nucleotides.